Dear juries, ladies and gentlemen, my dear colleagues, my respected supervisor and co-supervisor, and my other colleagues who are in online watching this defense, good afternoon. I am M.A.M. Siddique. I am as a Ph.D. student working from 2013. So I am actually third year Ph.D. students here under laboratory of reproductive physiology. The topics of my Ph.D. thesis is fertilization strategies for externally fertilizing fishes. Professor Otto Linhart is my supervisor, and Dr. Martin Senchka was my consultant during my PhD studies. So now I will briefly discuss about my thesis. In externally fertilization, there is three actors, mainly sperm, then eggs, and activation medium, and all have connected with each other. Without sperm, we cannot fertilize, or without egg, we cannot fertilize. We cannot do this, or even activation medium. If we keep eggs and sperm together, it will not be activated or fertilized. So these three are the main actors in a fertilization event. The conceptual framework for fertilization event is given in these figures. Here you can see that duration of egg motility when increase, and when the receptivity of egg when increase, then it increase the chance for fertilization. And here activating media is a mediator to help this event. So in my studies, mainly I have tried to work on the egg receptivity, and I use pre-incubation for three types of fishes. First on sturgeon, acipensarite, and then I work on sea bass, it's a marine teleost, and then a cyprinid eyed. So I will work with fresh water and also both in marine water, try to measure the duration of egg receptivity for these eggs, and try to see the effect of pre-incubation on this species. And beside that, sometime I included some more topics along with these topics to make it more informative. Suppose sometime I included egg to sperm ratio, or the manipulation of egg receptivity, or effect of sperm motility. So just include some more information to make this studies informative. So the main aims of my PhD thesis are to review the structure of sturgeon egg membrane and associated terminology, and secondly to study the duration of egg receptivity period and the effect of re-incubation of eggs in freshwater species. I worked on sturgeon, starlet, Acipensar ruthenus, and to examine the duration of egg receptivity period and the effect of pre-incubation of eggs in marine species. I used sea bass as a model species for this experiment, and try to understand the effect of activation medium to manipulate egg receptivity period for externally fertilizing fish. In this experiment, I use eyed as a model species, and finally try to develop a unique developmental mode, parthenogenesis, in Acipensar species, Acipensar ruthenus, starlet. So I will describe I will show some results of my recent papers. Now this is the review. I start my PhD work by reviewing the egg membrane structures, because I understood that it's very important to know to know about the layers and the envelopes of egg membranes, because it's very important. They have very important role in activation and fertilization mechanism. So I start to writing a review paper, and it already published in Journal of Applied Ethology. The main findings of this paper is I just described about the egg membranes layer of ovulated and fertilized eggs. These are ovulated eggs and these are fertilized eggs, and here the difference is this is inside the ovary, so there are also some thecal layer or basement or follicular layers, but when it released, then you will not see there's layers in the eggs. This is a fertilized eggs. After and you will see that cortical layers will be less because this cortical layer already dissolves and imbued with water and make very little in fluid. And I work a long time on these topics. I have several microscopic pictures from electron microscope, and we are planning to publish it slowly. And in this review, I describe standardize the terminology of different egg layers, because sometimes it's very confusing. The same layer, different author called it in different ways. Suppose the adhesive layer, some people said it a jelly layer, some people adhesive layer, some people say it jelly envelope, gelatinous envelope, external envelope. So it's confusing. When I was reading a lot of papers related to egg membranes, then that time I was totally confused. So I tried to standardize this terminology of different layers, and I hope this will be very useful for the future work on egg membranes or something. Then I described about the developmental stages of egg membranes and also the mechanism of polyspermy block in estrogen eggs. In estrogen eggs especially, there are four types of four mechanism of blocking the polyspermy. First one is the shape of the micropyle. This is called micropiles. Is sperm entry site is enough narrow so that only one sperm can penetrate through the micropyle. This is a mechanism of blocking polyspermy. Second one is after when first sperm reached there, after fertilization, then they form a fertilization cone, and it blocks the passage of sperm entry, and then other sperm cannot penetrate. It's also mechanism of blocking polyspermy. And the third one is the content of cortical granules of the micropyle canal. They filled up the micropyle canal and they block the way so that enter couldn't penetrate inside the sperm. So it's just after fertilization. So it's also a block of mechanism to block polyspermy. And the fourth one after complete formation of the vitelline space, you see that there is no only one cortical granules, because most of the cortical granules already dissolve and imbued with water and make perivitellin fluid. It goes over here and they secrete some enzymes inside the from the cortical granules and pigmented granules, and it makes a hardening of the egg envelopes. So sperm couldn't penetrate. So this is another mechanism of polyspermy block. So this is now I will discuss about some of my results of my research articles. It was my first paper from my PhD study, and here I tried to determine the effect of the incubation of eggs in fresh water in starlet and verifying sperm concentration of fertilization rate. And the main objective of this study are to estimate the adequate sperm to egg ratio for starlet fertilization, because we know that estrogen is the egg number of micropyle in estrogen is different. Here you can see the total 15 micropyle. Previous record was 13 micropyle. I got until 15 micropyle on the surface of the egg envelope. And this is the shape of egg micropyle canal. This is the micropyle canal. You can see there maybe fertilization cone. So when there is a lot of micropyle, there is a more chance of polyspermy. So the rules is if you put more sperm, then the chance of fertilization is higher. But in case of estrogen, if you put excess fertilization, there is a more chance to be polyspermy. and polyspermy egg cannot survive after a certain stage. So it's important to control the use of polyspermy and to estimate the optimum sperm to egg ratio for this species, and to minimize the sperm to egg ratio by modification of fertilization environment in order to save and prevent polyspermy. You know that when we strip the estrogen eggs, a lot of ovarian fluid are attached with the eggs, and sometimes it's very difficult to remove this ovarian fluid or seromic fluid from the eggs, and it's sticky, and sometimes it inhibits the sperm for motility or something. So it's I feel that it will be better to if we do some pre-incubation of eggs, so some of the ovarian fluid will be removed from the eggs and will facilitate the sperm and also eggs for fertilization. So it was my another objective. And this picture is a nice picture captured by transmission scanning electron microscope. Here one sperm is entering into the micropyle, and this is the flagellum, the tail of the sperm. I got this picture after 2 years work on electron microscope. And the main result of this experiment was we found that 430,000 sperm ratio, 430,000:1 ratio sperm, there is no effect of pre-incubation. Even 43,000:1 sperm don't have any significant effect, but there is a significant effect of pre-incubation on lower sperm to egg ratio. Here you see that there is significant effect on up to 1 minute. So that means that pre-incubation have significant effect when we use lower sperm to egg ratio. And there might be some question that why will pre-incubate, and if we pre-incubate, is there any possibility of activation of eggs? Then I checked some eggs. This is the 1 minute of this is after 1 minute of pre-incubation, unfertilized eggs, and this is 10 minutes of pre-incubation after 10 minutes of pre-incubation, unfertilized eggs. And here you can see this is the cortical layers. Here you will see that a lot of cortical granules over there, they are still unused. That means they are not going to initiate any cortical reaction. So this is actually not perivital in space. This is just some layer. This is the donor radiata. Here also the same. After 10 minutes of pre-incubation in water, eggs just activated, but they didn't go for any exocytosis. Here you'll see a lot of cortical granules. That means they didn't go for any reaction. Here you will see that after 60 seconds of post-fertilization, this sperm is just activated on water, but this is fertilized eggs. So you can see a lot of perivital space. There is no cortical granules because all the cortical granules dissolve with water, imbued with water after cortical reaction and make perivital space. You will see very big perivital space after 60 seconds of fertilization. So this is the difference. So that means that we can pre-incubate the eggs. There is no effect on activation or deterioration of egg quality for estrogen. And previous report was that Ginsburg said that estrogen can be eggs can be viable several hours in water. But I just proved by cutting some eggs on electron microscope. So the main findings of this study is up to 10 minutes after eggs activation in fresh water, estrogen eggs did not undergo cortical reaction, and without eggs pre-incubation, minimum effective sperm to egg ratio for starlet was estimated at 43,000:1 sperm to egg ratio, and after 0.5 minute of eggs pre-incubation in water, minimum effective sperm to egg ratio for starlet decreased at 430:1 sperm to egg ratio. Now I will move to the second experiment. I conducted this experiment in France, in Ifremer, with the help of Dr. Christian Fowell, my French colleague. And here we try to estimate the effect of pre-incubation, try to understand the duration of egg receptivity in seawater, and we also work in two types of egg, freshly ovulated eggs and overripe eggs. For sea bass, overripening is a very big problem, and eggs goes under overripening process very quickly within 30 minutes to 1 hour. And we try to pre-incubated all these eggs to see the effect of pre-incubation in this species. And the most interesting thing was the swelling process of sea bass. And when I was recording this video, I will show you a video. It was very interesting. You can see how fast they are swelling. I don't know how to start this. You can see the eggs are going to be round very fast. It's become oval shaped, and these are the lipid droplets, and they become they start swelling after activation, after contact with water, and within 10 to 20 seconds they swells. The main objective of this experiment was to determine the duration of egg receptivity in seawater, and another aim was to understand the effect of the incubation of sea bass eggs, both fresh and overripe eggs, on fertilization success. This is the main results of this experiment, and here you can see the effect of incubation for both freshwater, freshly ovulated eggs and overripe eggs. And here you can see that there is a big fluctuation of the fertilization rate after 3 minutes. And for the freshly ovulated eggs, and for overripe eggs also the same. After 3 minutes there is a big fluctuation. So it means that the duration of egg receptivity period for this species is 3 minutes, and is
moment last 5 minutes. Yeah, last 5 minutes.
Okay. But if I speak very fast, is any problem? I can speak very fast.
Yeah, this is what they are doing.
But I'm worried that you could understand or not. So it means that the duration of egg receptivity for this species for both the freshwater and overripe eggs are the same, 3 minutes. And here you will see the effect. They will compare the fresh and overripe eggs. And there are some photographs of swelling process of sea bass eggs. And the most interesting thing is in sturgeon, you can see the perivitellin space after fertilization by sperm. But here, without fertilization, you can see perivitellin space here. You see perivitellin space is forming without any fertilization after just activation in water. So different species have different behavior. It's a marine species, so their behavior is eggs behavior is a little bit different than sturgeon. So the main findings is duration of egg receptivity for sea bass is 3 minutes, and see it swells very quickly, 10 to 20 seconds upon activation in water, and all oocytes are not overripe in the same time, and it is very necessary to control the time of stripping, ovulation and everything, and we can get it's not that we cannot able to fertilize overripe eggs. It is possible to fertilize also overripe eggs after 1 hour, but it is only possible when eggs are collected immediately after ovulation. So we can wait until 1 hour. We can collect egg and fertilize within 1 hour. So next, third year genealogy paper, the main topics of this paper is to how to manipulate the duration of egg receptivity period by using of 3 different medium. I conducted this experiment in Poland with my colleagues Daniel Jarski and Slavek, and I already published this paper also. The main objective is to understand the effect of pre-incubation of eggs and activating medium on the percentage of egg embryos, and besides this we also try to investigate that whether there's activation medium has any effect on the sperm motility trait or not. So firstly we checked this and then we try to pre-incubate the eggs and try to estimate their duration of egg receptivity. There is some pictures of incubation of eggs in the jar. The main result of this experiment is when we use 3 different activation medium, fresh water, saline water and anaerobic solution, we found that in fresh water the duration of egg receptivity is 60 seconds. After that you will see that big fluctuation. And for saline water all are same, there is no significant difference among the treatments, and for anaerobic solution also the same, there is no significant too much significant differences. Little differences here, but not too much. So it means that for fresh water, the duration of receptivity is 1 minute, but for saline water and anaerobic solution, it can be changed. It could be until 2 minutes. So if the duration of receptivity we could increase 1 minute more, so there is a more chance of fertilization, because the longevity of sperm for this species is 45 to 1 minute. So if we could increase the duration of receptivity, there is more chance for fertilization. And there is another reason to using saline water and anaerobic solution, because they are also using as a desticking substances. And many hatcheries they use saline water or anaerobic solution to eliminate the stickiness of eggs. And this type of species is a cyprinid, and it's too sticky eggs, and that's why it will be best solution, saline water and anaerobic solution for this type of egg activation or fertilization. So main findings in duration of egg receptivity can be manipulated by activation medium, and we can find more activation medium, suitable activation medium for them. And saline water or anaerobic solution had no significant effect of selective sperm motility parameters. And finally, I documented the parthenogenesis in a Acivensis ruthenus species. Maybe my time is only 1 or 2 minutes. So this is the
exactly
unusual shape of parthenogenesis. I will not go detail for this. And the main thing is parthenogenetic eggs are usually mostly are 2 cells to 8 cells. After that they died off. And there is a relationship between the fertilization rate and the parthenogenetic activation rate. When the fertilization rate of the eggs is higher, then the parthenogenetic activation rate is lower. And especially when the eggs fertilization ability is low, then there is a trend to increase the parthenogenetic activation in eggs, especially for starlet. I just checked on starlet. And I documented this to understand that how we can differentiate between the parthenogenetically activated eggs and polyspermic fertilized eggs. So it's a very nice demonstration. And this is the, I already asked orally. So acknowledgement, first I would like to acknowledge my supervisor, Professor Ottomar Linhardt. That's why I am here. And my consultant, Jackie Koson, a lot of my lab mates, Marek Rodina, Boris Juba, Vladka, our lab technician, and Eva, and Jan Bart, my Danish, my colleague, Canadian colleague, is working on Denmark, and finally Lucy Kasirova. Without help, it was not possible to finish this PhD within 3 years, and he always supports a lot. This is the this is the projects financially supported, a lot of projects. And finally the curriculum vitae, I was graduated in marine science, not in fisheries, and after that I did second masters from University of Tromsø in fisheries, aquaculture, economics and management, and after that I joined here as a PhD student. I have published more than 10 papers.
It's not necessary. You can read it.
Okay.
Yeah.
And finally, thank you all for your attention.